So I once again thankful to the Academic Staff College for giving an opportunity. So let us start I would like to give brief about the environmental pollution. So what are the important pollution which are taking place in the environment? And it's brief about that. After that, I'll concentrate on the control strategies, particularly on bioremediation of environmental pollutants. You know that pollution is the introduction of harmful substance or products into the environment, particularly to water, air, and land. So it is called as the pollution, the definition of pollution. So in this, some of the pictures, I would like to show you the extent of pollution we are doing with respect to water, soil, and air. I'll go briefly to show that some of the pictures. You can see that how the water is polluted by releasing the pollutants into the water bodies. So you can see this in these pictures. So particularly this water pollution is one of the important pollution. There are two types of pollution, water pollution, categorized into two types. One is point source and non-point source. So you know that point source are the easiest to identify and control, and non-point source are the ambiguously defined and harder to control. These are the mainly classified into two types of pollutant, that is a point and non-point source of pollution. So you can see that what are the important point source of pollutions. It includes the products from factories, waste products from factories, waste from sewages system, waste from power plants, water from underground coal mines, waste from oil wells, etc. These are all considered as the point source of pollution. Non-point source of pollution includes the term non-point source encompasses a large range of sources, such as when rain or snow moves through the ground and picks up pollutants as it moves towards a major water body. So the agricultural runoff, which carries the fertilizers and pesticides and enter into the downstream body. So that is called as point source of pollution. So in this, you can see that the pictures of non-point source of pollution. So let us and some of the pictures of environmental pollution, particularly air pollution, how we are releasing polluting the air. So the main source of air pollution is the release of carbon dioxide into the atmosphere. This happens because of deforestation and burning of fossil fuel, etc. And sulfur dioxide is another air polluter, and is released into the atmosphere by the burning of sulfur-containing compounds of fossil fuels. So you know that the importance of air pollution, particularly during the December and January month every year. You can see that Delhi is one of the most worst affected city by the air pollution. So you know that what are the reason for that. That is the causes of air pollution. So chlorofluorocarbons are major contributors of air pollution. So these are all the things which you already know, that just I want to give the brief about the environmental pollution. And then consequences of air pollution also. So you know that is a good carbon dioxide is a good transmitter of sunlight, but it also partially restricts infrared radiation going back from the earth into the space, which produces the so-called greenhouse effect that prevents the drastic cooling of the earth during the night. So that is called as global warming. Increasing the amount of carbon dioxide in the atmosphere reinforce this effect and is expected to result in a warming of the earth's surface, that is called as global warming. Everybody knows that. So like that, in the land, how we are polluting the land also, you can see that in these pictures. Some of the pictures I want to show you. So main causes of land pollution includes the construction waste, agriculture waste, domestic waste, and industrial waste, which we are dumping on the land without proper disposal. That causes the land pollution. So construction waste also, building take up the resource of land, trees, and chop down and used to make buildings, etc. And agriculture is another important polluter of soil. The modern agriculture, we are adding millions of tons of pesticides, herbicides, fertilizers, and manures in order to increase the crop yield. So that causes the land pollution. And domestic waste also, you can see in this picture how we are dumping the waste on the soil and causing the land pollution. So you can see that industrial waste also produces plastic factories, chemical plants, oil refiners, nuclear waste disposal. So these are all the things which are responsible for this environmental pollution. Consequences of land pollution also, you know that how it affects the land pollution, it affects the wildlife, and also vegetation that provides the food and shelter is destroyed. Land pollution can seriously disrupt the balance of nature, and in extreme causes can cause human fatalities also. So pesticides can damage crops, kill vegetation, and poison the birds, animal species, etc. So these are all the consequences of land pollution. So I would like to brief about what are the environmental contaminants, particularly if you take the chemical compounds, which are very important for considered as important pollutants, are the natural as well as xenobiotics also. Natural occurring compounds in the environment that are present in unnaturally high concentration, example, crude oil, refined oil, phosphate, etc., heavy metals. And xenobiotics are the chemicals that are synthesized compounds that are never occurred in the nature, example, pesticides, herbicides, plastics, etc. So these are the important pollutants which are causing the environmental pollution. So, and also in addition to that, some of the hazardous pollutants also, we consider them as very hazardous. The misplaced chemicals that causes harmful effects in the environment, they includes the toxic compounds, carcinogenic, mutagenic, teratogenic, flammable, corrosive. So these are the harmful effects of some of the important chemical compounds in the environment. So some of the petrochemical compounds and heavy metals that includes the mercury, lead, cadmium, chromium, zinc, PCBs. These are all some of the common pollutants which are causing the pollution in the environment. So this is the table. You can see that important contaminants, common pollutants in the environment. Okay, like that. Okay, so this is about the just brief about the pollution, which you already know. Just I wanted to show that the pollution of the air, water, and soil. Now come to the conventional methods of remediation. What are the methods which are practiced in order to control these pollutants or to reduce this pollution? Pollutants includes the dig up and remove it into a landfill. Either what we are doing is normally dig up and remove it to landfill, or cap and contain. These are the two methods which are conventional methods. Maintain it in the same land but isolate it. So these are the fundamentally two important methods which are doing. What are the problem of these two methods? Risk of excavation, handling, and transport of hazardous materials, and very expensive to find another land to finally dispose these materials. So this is the drawback of these methods. And the cap and contain means maintain the pollutants in the same land but in isolated manner. Only an interim solution. It is not a permanent solution because the pollutants remains in the same form in the environment. It also requires monitoring and maintenance of isolation barriers for a long time. These are the conventional methods which are there. So products are not converted into harmless products, stay as threat. So by these two methods, the products will not be converted into harmless, but they will remain the same form in the environment. So what are the better approach to these conventional methods? So what are let's see, better approaches in order to control this pollution or prevent this pollution. What we have to do is either to destroy them completely if possible, or transform them into harmless substances. So these are the better approaches that we can do in order to control the environmental pollution. Destroy means completely we can destroy that pollutant by various methods if it is possible. Otherwise, we have to transform the harmful substance into the harmless substance. These are the approaches which we have to do. So methods already in use are high These two methods includes the methods already which we are following or practicing are high temperature incineration. That is to destroy the pollutants. What we have to do is either we have to go for high temperature incineration, the burning of the pollutants in the environment. And then chemical decomposition like chlorination, UV oxidation. These are the two methods which are practiced in order to destroy the pollutants, environmental pollutants. So like incineration and chemical decomposition using hydrochlorination, dechlorination, or UV oxidation, etc. So, but are these methods what we are doing are the effect? What are they effective? We have to see that at what extent it is good by burning of the pollutants, by using high temperature. Is it good or bad? So all these things which we have to see. So again, you can see that to some extent can do the practice these methods, but it is not the complete solution. And there are many drawbacks of these methods. What are the, let us see, what are the drawbacks of these conventional methods of these technological complexity? We have to go for a technologically complex method. The cost for small scale application is very expensive. Lack of public acceptance, especially an incineration, that is burning of the pollutants in the environment, is not acceptable because incineration generates more toxic compounds than the original compounds. Materials released from the imperfect incineration causes undesirable imbalance in the atmosphere. Example, ozone depletion and fall back on the earth and pollutants. So same other environment. Dioxin production, or particularly burning of plastic, which releases the dioxin, which is known for the as a carcinogen, leads to cancer. So let us see one of the important methods that to overcome all these problems. What is the methods which we have to go for or in order to control the pollution? So this bioremediation is one of the important emerging field area in order to control the pollution or remove the pollutants from the environment. Bioremediation makes an effective, better approach possible. So in this method, either by destroying or rendering them harmless, the pollutants using natural biological activity. Here, in the earlier previous slides, we have seen that only the physical and chemical methods are used in order to control the pollutants. But the bioremediation makes an effective, better approach. The principle of bioremediation is that either by destroying or rendering them harmless using the natural biological activity. That is the method of this bioremediation. So let us see what is bioremediation. Bioremediation is let us see the definition of bioremediation. Is a use of living organism to transform, destroy, or immobilize the contaminants. So that is the definition of bioremediation. The ultimate goal of this bioremediation is to detoxification of the parent compounds and conversion of products that are no longer hazardous to human health and environment. That is the ultimate goal of this bioremediation. That is to convert detoxification of the parent compounds and conversion to products that are no longer hazardous. So that is the ultimate goal of this bioremediation. So what are the things which you can do by using bioremediation? What needs to be clean up? Soil, water, and air, and sediments can be cleaned up using this method, bioremediation method. Pollutants enter environment in many ways, different ways. That already I have explained about the pollution, how this environment is polluted, either by tanker spills, truck accident, ruptured chemical tanks, industrial sites. So various ways that environment is polluted, so that can be cleaned up by using this bioremediation methods. So bioremediation is one of the important nowadays the emerging field of pollution control. So here the bioremediation is broadly can be classified into two types. So that is in situ bioremediation and ex situ bioremediation. So what is in situ bioremediation? So I'll tell you later. So that includes various methods in situ bioremediation, bioventing, biodegradation, biostimulation, biosparging, bioaugmentation, and natural nutrition. Like that, in ex situ bioremediation, that includes land farming, composting, biopills, and bioreactors. Let us see what is in situ and ex situ bioremediation. In situ bioremediation is one of the most important method of bioremediation, where the pollutants are is polluted in their place. In that place only, we are doing all this treatment process. So we are not taking that pollutants to the outside from the original place. So in the place where it is polluted, we are performing this remediation process that is called as in situ bioremediation. So in this in situ bioremediation, there are various methods on there. Includes bioventing, one of the most common approach in soil, and which includes the supply of air and nutrients via wells that takes advantage of indigenous microorganisms. Here, the bioventing means we are supplying the air or nutrients to the polluted place, polluted environment, polluted soil or water, where the microorganisms which are already present in that will start multiplying and growing, so that perform the degradation process that is called as bioventing. And in biodegradation process, supply and air and nutrients also by circulating aqueous solutions through the contaminated soil or groundwater. And another method is biosparging, that is injection of air below the water table, where if there is a water table or groundwater pollution is there. So in that cases, we can increase the groundwater oxygen concentration and mixing in saturated zones and so on, so that increase the growth of microorganisms that decompose the pollutants. And another method is the bioaugmentation, that is addition of indigenous or exogenous. Here, exogenous microorganism means which are not present inside the polluted place. We can bring and add the microorganisms which are known to degrade the pollutants very effectively, or potential microorganism isolates. We can use that. So that is called augmentation, addition of the microorganisms in addition to the indigenous microorganisms. So that is called bioaugmentation. And biostimulation, I already explained. The stimulation means stimulate the microorganisms to grow in the polluted land. So here in this picture, you can see how this bioventing is done with the schematic diagram. So vacuum pumps and used to feed the nutrients and moisture to the groundwater, particularly polluted, is a process of stimulating the natural in situ biodegradation of contaminants in soil by providing air or oxygen to existing soil microorganisms. So what I already explained, you can see in the schematic diagram. The oxygen is the most commonly supplied through the direct or air injection into residual contamination in soil. In addition to degradation of adsorbed fuel residuals, volatile compounds are biodegraded as various vapors move slowly through the biologically active cells. So that is the mechanism of the biodegradation in the bioventing method. So another method is the in situ bioremediation is the biosparging. That is, in biosparging, air or oxygen and nutrients, if needed, are injected into the saturated zones to increase the biological activity of the indigenous microorganisms. Here you can see that in this picture, the biosparging can be used to reduce concentration of petroleum constituents that are dissolved in the groundwater. If there is the pollution of groundwater with by petroleum compounds adsorbed to the soil below the water table and within the capillary fringes, we can perform this method. And in addition to that, natural attenuation also takes place using the biodegradative capacity of indigenous microbes without additional enhancement So that is by naturally also. So if you give these microorganisms also perform this function. So what are the steps involved in the bioremediation? That includes the site investigation, treatability studies, recovery of pre-compound products, and removal of the contamination source. So these are some of the steps which are involved in the bioremediation method. In the same way, the another method of bioremediation is the ex situ bioremediation. I told you ex situ bioremediation is nothing but the performance of bioremediation from other than the polluted land. So we can take out that pollutants to some other place and perform this remediation process. That is called ex situ bioremediation. That includes the various methods popularly known as the land farming. That term itself explains land farming. Here, the contaminated soil is excavated and spread over land, and soil is periodically tilted to improve the aeration. For example, if the soil is polluted, so in a particular place where we cannot perform the in situ bioremediation, so you can take out that pollutant to some other place where it can spread over the barren land, so that where it performs the naturally, so you can see the degradation of pollutants. The remediation due to indigenous microorganisms as well as chemical and physical process also. It is generally limited to the superficial of 10 to 35 centimeter of soil, can reduce the monitoring and maintenance cost. And another method is the ex situ bioremediation is composting. It's a very common method of ex situ bioremediation. So it combines contaminated soil with non-hazardous organic amendments like manure and agriculture waste. So you can use this waste by particularly biodegradable waste. So you can perform this composting. So ultimately you can get the compost which is used for the manure for agriculture production. And another method is biofuels. Is a combination of land farming and composting is together is known as biofuels. And it controls physical loss of contaminants. And another important method of ex situ bioremediation is the bioreactors. You know what are bioreactors? Bioreactors are the fabricated or engineered structure where all the environmental factors which are required for the degradation of pollutants are controlled. So you can control that all these environmental factors are like temperature, moisture, pH, anything can be controlled, monitored. So it is a fabricated and engineered structure. So in order to perform the bioremediation process. So here soil and water pumped up from a contaminated plume and processed through an engineered containment system. So that is called bioreactor. So here the degradation in bioreactor is generally greater than in situ because the contained environment is more controllable and predictable. Here, what is required for the fast degradation of the pollutants can be known and accelerate the process. So that is the advantage of these bioreactors. So in the ex situ bioremediation in this picture, you can see that how the excavated soil is taken into some other place and spread over the land, that is called. And also you can see that different types of bioreactors which are used for the degradation of pollutants. So slurry phase bioreactor and solid phase bioremediation, that is using the slurry phase. If it is a pollutant is in the slurry form, we can use this type of bioreactors. And if it is in the solid state, so you can see that soil can be converted into compost by composting method, land farming, and biofuels. So these are the fundamental methods of ex situ bioremediation. So in this picture also, you can see that how this land farming and composting can be done. A thin layer of excavated contaminated soil is spread over the land so that it be converted into non-hazardous or decomposition of the pollutants by microorganisms. Here, contaminated soil is excavated and spread over land. Soil is periodically tilted to improve the aeration. and remediation due to indigenous microorganisms as well as chemical and physical process generally limited to the superficial of the 10 to 35 centimeter of soil. So here also you can see the composting. There are various methods composting has been used to degrade the solid waste materials such as leaf litter, waste, sewage sludge, and food waste, etc. Okay. So here also biofuels are the system offer the potential for cost-effective treatment of contaminated soil. Like composting, biofuels provides favorable environment for the indigenous microorganisms to degrade the contaminants present in the soil matrix. So the reactors, bioreactors are the advanced containing containers or the engineered structure. So here you can see different types of bioreactors, slurry bioreactors. Is where a slurry bioreactor may be defined as an containment vessel and operators used to create three phases, that is solid, liquid, and gas mixing condition as to hasten the biodegradation of soil-bound water and water-soluble contamination as a water slurry of the contaminated soil. So here the sediment or sludge and biomass are usually used for this degradation process. What are the advantages of this bioreactor includes it is enhanced process control, faster rate of biodegradation of contaminants are possible, better physical contact between the pollutants and microorganisms. That is the most important thing. In the bioreactors, the microorganisms will directly come in contact with the pollutants. So that is why the process will be more fast and effective. And distribution of nutrients, gases, and other materials for support of biological process is greatly improved. So that is a thing which can be achieved by these bioreactors. And optimal soil, sediment, or sludge particle size can be selected. So you can select the optimal soil, sediment, etc. in this method. And liquid phase organic solubilities may be enhanced by surfactants application. Surfactant can also be used in this method also. So there are different types of bioreactors are there. Fixed film bioreactors and fixed film. So here these are the advanced bioreactors where fixed film bioreactors have become conventional technology for treating biodegradable contaminants in air and water also. Then some suspended growth bioreactors are there. So here completely mixed activated sludge systems, continuous wastewater, plug flow activated sludge system, sequence batch reactors, batch wastewater, and anaerobic digesters. These are the different types in the suspended growth bioreactors, which are very advanced. The excess biomass is wasted to sludge disposal process. So these are the different bioreactors. Let us see what are the feasibility of this effectiveness. What are the important factors which are important in the bioremediation process? How this works? What is the mechanism? Let us see. A function of it depends on any bioremediation process. It depends on the microorganisms. What are the microorganisms which are present in the soil and isolated soil, and environmental factors and type of contaminant and its state. So all the three are very important to achieve, in order to achieve the maximum bioremediation process or degradation process. Say for example, environmental conditions includes pH, redox potential, temperature, moisture, nutrients. These are all comes under the environmental conditions. Okay, and then compound structure, whether that compound can be degraded easily by the microorganisms depends on the type of compound, its structure, and properties of the compound. And then microorganisms. What are the microorganisms which are present in that? Whether they are potential in order to degrade that process. So in this diagram, so you can see that the feasibility and effectiveness depends on the microorganisms environmental factors, and contaminant type and state. Let us see some of the important examples are the microorganisms which are used in the degradation of pollutants. So particularly aerobic bacteria. Example, Pseudomonas alcalgens, Spingomonas rhodococcus, and Mycobacterium, which shown to degrade pesticides and hydrocarbons, alkenes, and polyaromatics. Many may be able to use the contaminant as a sole source of carbon and energy as energy source. And some of the other bacteria, microorganisms includes the methanotropes. So these are the aerobic bacteria that utilize methane for carbon and energy. So that's why it is called as methanotropes. Methane monooxygenase has a broad substrate range. Here they produces an enzyme called a methane monooxygenase, an enzyme produced by these organisms. They active against wide range of compounds. These methanotropes, example, chlorinated aliphatics such as trichloroethane and 1,2-dichloroethane. And other microorganisms includes anaerobic bacteria, which are not used as frequently as aerobic bacteria, but are also used, can often be applied to bioremediation process of polychlorinated biphenyls, commonly known as PCBs, in river sediments, particularly in river sediments, trichloroethane, TCE, and chloroforms. These are the compounds which are degraded by the anaerobic bacteria. And in addition to the bacteria, anaerobic, aerobic, and methanotropes, some of the fungi are also known to degrade the pollutants, are able to degrade the diverse range of pesticides, persistent or toxic environmental pollutants. So let us see how these microorganisms use this contaminant and convert that contaminant to harmless or decompose that contaminants. So contaminants may serve as a primary substrate. These microorganisms may use these pollutants as a primary substrate, enough available to be the sole energy source, or secondary substrate to provide energy not available in high enough concentration. And also they use it as a co-metabolic substrate. That means fructuous transformation of compound by microbe relying on some other primary substrate. That is why co-metabolite. So the co-metabolic process substrates used as a, in addition to the other primary substrates also. You can see in this requirements for microbial growth, how they grow the microorganisms using these pollutants. So toxicants, either as a carbon source or energy source they use. So here, what is required is electron accept or oxygen, nitro, or sulfur, NO3 or SO4, etc. are required. That is called electron acceptor. And environmental condition which influence this chemical process, are the conditions of the temperature, pH, and electrical conductivity, and nutrients also required. That is nitrogen and phosphorus, and trace elements also. So that's the requirements for microbial growth in the polluted feed. So here you can see that what happens in this process is electron exchange will take place. Oxygen and carbon are energy source, and the donor, carbon dioxide and water are the released with the help of the electron transfer takes place. So here you can see that both aerobic as well as anaerobic process are the which takes place in this bioremediation process. If the oxygen is a terminal electron acceptor, the process is called as aerobic biodegradation process. All other biological degradation processes are classified as anaerobic biodegradation. In most cases, bacteria can only use some terminal electron acceptor. Facultative aerobes use oxygen, but can switch to nitrate in the absence of oxygen. So that's why they are called as facultative aerobes. So that is the reason. So let us see some of the bacterial metabolism of the takes place. Oxidation, co-metabolism, anaerobic, which takes place through denitrification, manganese reduction, iron reduction, sulfate reduction, and methanogenesis. So these are the process which takes place in the during the degradation. So co-metabolism is a bacterium uses some other carbon and energy source to partially degrade contaminants. So that's why it is called co-metabolism. Organic aromatic ring compound, example pesticides, aliphatic and aromatic, using the it is called as co-metabolism. So some of the examples I would like to give: electron transfer to oxygen as electron acceptor. Here you can see the benzene. If you take one of the important pollutant, a very carcinogenic compound, how it is converted into carbon dioxide and water, and plus biomass, increased number of bacteria by using anaerobic degradation, biodegradation process. Here you can see the chemical process which takes place. The electron transfer to nitrate as electron acceptor. Nitrate is and carbon dioxide and nitrogen, nitrate and water and biomass. So that is the mechanism of degradation of organic chemical, particularly benzene. Some of the xenobiotic compounds have been produced artificially. Okay, that is not. I'll go to the other examples. So let us see how this biodegradation of petroleum compounds, particularly the petroleum compounds, are the very important compounds which are responsible for the oil pollution, particularly in the ocean. The oil pollution is one of the important pollution which affect the ocean or the marine environment. Compounds are categorized in two groups: aliphatic hydrocarbons and aromatic hydrocarbons. Here the enzymes produced by the microorganisms like monooxygenase and dioxygenase, which are responsible for this biodegradation of petroleum compounds. So in this also you can see that some of the straight chain aliphatic hydrocarbons compounds, how they are degraded by the organisms. Cyclic aliphatic hydrocarbons also. Let us see some of the important microorganisms involved in the biodegradation of xenobiotic compounds. Xenobiotic compounds are the compounds which are produced by our not present naturally. They are synthetic compounds. Okay, some of the Archibacter, Chromobacter, alkanogens, Acinobacter, Azotobacter, Pseudomonas putida, Candida, Trophilicus, etc. Aspergillus. Some of the fungi also known to degrade these compounds like Aspergillus, Penicillium, etc. Like E. coli also, some of the bacteria like E. coli, surfactants, some alkaloids, Chromobacter, pesticides are also degraded by some of the Pseudomonas aeruginosa, etc., and Arthrobacter species, and Parathion, another important pesticide which is degraded by the Pseudomonas species, and E. coli also. And genetically, how this genetically modified microorganisms are also used in the degradation process. I'll tell you later what is that. But okay, so how to measure this? So assay to degradation ability. How we can say that these microorganisms degrade these pollutants? That is why we have to go for the assay system for degradability test. How to conclude that these microorganisms are degrading the pollutants? That's why we have to go for the biodegradability test. So that is measurement of oxygen consumption by nanometric and electrolytic system, measurement of carbon dioxide evolution by the infrared methods, chemical methods, and use of radio-labeled substrates, measurement of disappearance of the chemicals by GC, determination of the reduction of DOC, and chemical biodegradability under anaerobic conditions. So these are all the assays which are used in order to confirm whether the pollutant is degraded by the microorganisms or not. So like a measurement of oxygen consumption, how much oxygen is consumed. So that also we can measure by manometric and electrolytic system, measurement of carbon dioxide evolution by infrared chemical methods, and use of radio-labeled substrate, that means incorporation of radio-labeled substance to the pollutants, so that can detect, trace that elements by radio-labeled elements and during different stages. And also measurement of the disappearance of the chemicals by gas chromatographic method also we can confirm. Like that, there are other methods also. So let us come to the application of genetically engineered strains to clean up the. So far, what I explained are the naturally occurring microorganisms which are used for this bioremediation process. Here, the one of the important example, which is popularly known as the superbug, which is genetically engineered microorganisms, which is used for the cleanup of the particularly oil pollution. It is created in 1970s itself, not as a new. It's a old. Isolated strains of Pseudomonas from contaminated soil and contains plasmid that encode genes for breaking down the pollutants. What they have done is they genetically modified this Pseudomonas putida so that it can very effectively degrade this oil pollution. So that is what is it called as petroleum eating bacteria. It's also called as superbug. Okay. So here, and some of the other microorganisms also used, genetically modified microorganisms, which are used as E. coli to clean up heavy metals like including copper, lead, cadmium, chromium, and mercury, and biosins of bacteria capable of detecting variety of environmental pollutants. Then here also how this genetic engineering bacteria to remove the metals from the environment, how they are synthesized, genetically modified. So what are the important factors which are required for the removal of these pollutants? Includes by microorganisms, nutrient availability, environmental condition, and metal content also. So environmental condition affecting the degradation, particularly conditions required for microbial activity and optimal value for degradation. So you can this table, you can see that what are the important parameters which are required for the degradation of pollutants like soil moisture, soil pH, oxygen content, nutrient, temperature, and contaminants, heavy metal, type of soil, etc., and conditions required for this microbial activity and optimal value for an oil degradation. Particularly if you take oil degradation, what is the optimal value required? For example, soil moisture, what is required for this, and soil pH and oxygen content, carbon nutrient level. So these are all the optimal value which are required for the effective degradation of pollutants. In the what I explained is so far as the microorganisms which are commonly used, that is called microbial degradation or microbial bioremediation. In the same way, like microorganism, we can also use plants for this bioremediation process. That is called as phytoremediation. Now, what is phytoremediation? Phytoremediation is nothing but the use of plants for the removal of the pollutants in the environment. Okay, there are various methods in the phytoremediation, which includes phytoextraction or phytoaccumulation, where plants used to accumulate the contaminants in roots and above ground biomass. So plants where that has ability to absorb the pollutants and it will be accumulated in the plant above ground level, above ground mass, that includes the stem, root, stem and leaf, flower, etc. So pollutants is going to accumulate in that. That's why it is called as phytoextraction or phytoaccumulation. The extracted pollutants can be accumulated pollutants can be extracted from the plant. So that's why it is called as phyto-extraction or phyto-accumulation. And phytotransformation or phytodegradation is there. That is, the term itself explains that phytotransformation means uptake of contaminants and transformation of to more stable, less toxic or less mobile forms. That means once the plant absorb the pollutants from the soil, that plant can have the ability to transform that pollutants, that highly toxic pollutant, to more stable and less toxic or less mobile forms. That's why so you can reduce the impact of pollution, soil pollution in the soil pollution by the using some selected plants. So that is example. Metal chromium can be reduced from hexavalent to less mobile, non-carcinogenic trivalent. You know, hexavalent chromium is more toxic. Hexavalent chromium is more the very carcinogenic and toxic form of chromium. So it can be converted into trivalent, which is not non-carcinogenic form of chromium. So you know, chromium is one of the important pollutant which is used in the industry, particularly chromium plating is one of the important industrial industry application of this chromium where it is used, so that can be removed from the soil using this phytotransformation or phytodegradation method. And another method is the phytostabilization. That is the mobility and migration of contaminants are reduced through the sorption into the plant. Sorption means absorption. Stabilize, they'll stabilize in the plant, the pollutants are, okay. The same way, another method is the rhizodegradation. Rhizo means roots. So the pollutants which are present in the soil can be degraded or absorbed by the roots of the plants, so that the root system, they have the ability to break down of contaminants through the activity of rhizosphere. You know, rhizosphere is the place of surrounding the roots. That's why rhizosphere, where that in addition the microorganisms are also present in that rhizosphere. That also helps in the degradation of the pollutants in the plant system. And rhizofiltration, here water remediation techniques here used to reduce the contamination in natural wetlands and estuaries areas. That's what rhizofiltration, where the plants grown in the water wetlands and estuaries. This technique can be very effective. So that is why it's called a rhizofiltration.
So what is the difference between the phytoaccumulation and phytostabilization? They are literally phytoextraction and phytostabilization. They literally
Phytoextraction, you can extract that pollutants. The stabilization, it cannot be extracted. It will be present in the without affecting. It will be present stable in that plant. So accumulation means in the large quantity is going to accumulate in that, so that you can extract from the plant. The pollutant can be extracted from the plant.
Okay, thank you, sir.
Okay. So in this picture, you can see that all the what I explained so far, all the process can be given diagrammatically represented in this picture. So in this picture, you can see that phytoextraction, where that is accumulated material pollutants can see, and phytodegradation, using roots. So all these are represented in this picture. Can you see? So the all the process are given represented in this picture. So you can see that how this pollutant stabilization, phytostimulation, phytoextraction, phytodegradation. In addition to that, one more is the photovolatilization is also. Volatilization means the pollutants which are absorbed to the plant in through the leaves can be volatilized through the various processes like transpiration and all those things. Some of the compounds can be converted and volatilized. So that is also one of the important process. Okay. 1 minute. ಒಂದು ನಿಮಿಷ ಹಾಗೆ ಇರು. ಸರ್ ನಿನ್ ನಂಬರ್ ಬೇಕಿತ್ತು ಬರುತ್ತೆ. Sorry for the interruption. Okay, so in this picture you can see the same process what I have explained so far. Okay, so non-biological remediation technology and phytoremediation are not mutually exclusive. Okay, so here you can see that some of the pollutants which are done through using this and then what are the advantages of this? Let's see. It is a cost effective when compared to other methods and conventional methods. Natural process, nature method, more aesthetically pleasing, minimal land disturbance, reduce potential for transport of contaminants by wind, reduces soil erosion. Hyperaccumulators of contaminants mean a much smaller volume of toxic waste. Multiple contaminants can be removed with the same plants. So those are the advantages of this phytoremediation. There are some disadvantages also, like slow rate. So any natural process is a slower process, unlike the chemical or physical method. So the biological methods are all very slow process. Okay, and slower and difficult to achieve acceptable levels of decontamination. Potential phase of transfer of contaminants. Sometimes contaminants also convert into another contaminant. Possibility of contaminated plants entering the food chain. So sometimes the food chain, these plants, the cross pollination of the or the contamination of these plants with the food plants also, food chain also. Possible spread of contaminant through falling of leaves against. So these are some of the disadvantages of this phytoremediation. And also decrease in action during winter months, and trees and plants require care. Contaminant might kill the tree. Sometimes the plants are, if there is high concentration of pollutants, the plants itself will be killed. Degradation products could be worse than the sometimes original compounds. Much testing is needed to before a procedure can be utilized. So these are some of the disadvantages. So example, I'll give you some more example and very successful stories of this phytoremediation or bioremediation. In the 20th most common element is the earth crust is the, and 12th most common element is the human body is the arsenic. Is a major worldwide contaminant and can arise through the industrial activity, like including pesticides, mining, combustion, etc., or from soil and groundwater, associated with acute poisoning and linked to liver, lung, kidney, bladder, cancer, etc. So these are some of the impacts of this arsenic. Okay, so this can be remediated by this biological method, that is using in India, particularly in around the risk of this bioaccumulation of arsenic in water bodies. Okay, that is 40 million people drink well water containing toxic level of arsenic. So this a plant which is used accumulation of arsenic using plant, okay, particularly using phytoremediation process. So this has been excellent removal of this arsenic from the using some of the important plant, particularly phytoremediation process. Here you can see this arsenic as hyperaccumulation in the bacterial absorbance reduces. Bacterial some of the strategies behind this cloning. This is the genetically engineered microorganisms or plants as a bioaccumulation of arsenic. So here you can see that arsenic using the prokaryotic arsenic genetically modified plants which are used for this accumulation of this arsenic. Here also you can see that testing of any doubts? Hello? Hello?
No, sir, no doubts.
No, sir.
Continue.
So this you can see picture, you can see that some of the assays in order to test that expression of the pollutants, the tracing of the degradation of methods what I explained so far. So this plant system, so you can detect the pollutants, okay, how this degradation process takes place. You can see that some of the radio bypass carbon assays which are used for this testing of. So some of the examples I would like to give are the arsenic contaminant by phytoremediation, particularly the Teres vitata is one of the important plant species which has natural arsenic hypoaccumulator, which is known as naturally arsenic hyperaccumulator of arsenic. So in this picture, you can see that the plant can tolerate soil concentration of up to 1,500 micrograms arsenic per gram and can be accumulate up to 23 milligrams arsenic per gram in its shoots. So that is the extent so that can be used for the degradation, the extraction of this particularly arsenic compound. The striking difference between the Teres vitata and arsenic non-accumulators is the enormous transport of arsenic from the roots and shoots. So enormous transfer, that is very effectively it will absorb the arsenic. In most plants, only a small fractions of the arsenic taken up from the soil by root accumulators in the above grounds. But whereas you can see that Teres vitata accumulates up to 95% of the arsenic in the above ground tissues. That's why it is very useful plant species which is used for the phyto extraction of arsenic in the arsenic contaminated soil. It is also called as the Chinese ladder fern. It is also known as Chinese ladder fern. Teres vitata is also known as the brake fern, is a highly efficient accumulator of arsenic. So here also given some of the name plant species which are used for the phytoremediation process: Eichhornia, Thalpsi, and then very important acro species. I could okay, like various species, plant species which are used for accumulation of various compounds and the concentration, how much it can accumulate in the plants their system. So you can see this in this picture. So one more example I would like to give is the potential water of water hyacinth. Commonly, you know that it is a very common plant species which grow in the polluted water bodies, particularly. Okay, it is also used to remove a phosphorus pesticide, the ethion, where for investigated the disappearance of rate of constants of the ethion, you implied that plant uptake and phytodegradation contributes 69% and that of microbial degradation up to 12%. So this plant can be used as a one of the important species for the phytoremediation process, which can remove some of the pesticides in the wood. Okay, the accumulated ethion in live water hyacinth plant decreased by 55 to 91% in the shoot and 71 to 81% in the root system after the plant growing in 1 week in ethion-free culture. So that's the effective plant species which is used for this phytoremediation. So some of the other methods are also there. A mass of balance of field trial for carbon tetrachloride phytoremediation using poplar, another plant species. For phytodegradation is the likely okay. It's another plant species which is used. So another plant species very commonly used as the pig slurry cleanup, that is using duckweed, lemma, the plant species which can be used as in order to remove the nutrients, excess of nutrients, where excess of nutrients in the water body leads to eutrophication. So this plant duckweed can be used in order to remove the excess of nutrients in the water bodies. So in this picture, you can see that rhizofiltration using this sewage and urban waste waters, halogenated residues. Okay, so intense root system where the plants which produces a massive root system can also be used for this phytoremediation process. So some of the explosives are also removed from the soil using various plant species. You can see that explosives, RDX, TNT, are also degraded by or used by the plant species. Another important plant species is Salicornia. Salicornia, this is also known as pickleweed, which accumulates salt in vacuoles, a form of table salt that can be extracted from the plant. So where there is salinity is more, if you grow these plants, so that will remove the excess of saline in the soil. So that is also another method of bioremediation process, particularly using plant species. So here, water bodies, in the case of wastewater contamination, also can be done by this plant species. Cleanup strategies are already explained. Okay. Let us come to the advantages and disadvantages of bioremediation process. Okay. What are the advantages of bioremediation? Either it may be plant or microorganisms. Is a natural process and is therefore pursued by the public, and is also useful for complete destruction of wide variety of contaminants. So wide variety of contaminants can be degraded by this bioremediation. Instead of transforming contaminants from one environmental medium to another, for example, from land to water, air, the complete destruction of target pollutant is possible in this bioremediation process. Okay, and also can be carried out on site. That I already explained. What is on site bioremediation? That is in situ bioremediation without causing a major disruption of normal activity. And also bioremediation can prove less expensive than the other technologies that are used for cleanup of hazardous waste. And also can be highly specific sometimes. Okay, if mineralization occurs, get complete degradation and cleanup. The process is the bioremediation process is a very simple process. Okay, is also limited. There are some disadvantages also. It's not a complete solution, not a solution for physical chemical methods, because it has its own limitations. So it is limited to those compounds that are biodegradable easily. Microorganisms cannot degrade all the compounds. Only biodegradable materials can be degraded by these microorganisms, or not all compounds are susceptible to rapid and complete degradation. There are some concerns that products of biodegradation may be more persistent or toxic than parent compound. It is not always, but some exception. Sometimes the the degraded compound, which is produced by degradation of microorganism, is more toxic than the original parent compound. Possibilities are there, so not always. So biological processes are often highly specific microbial pollution populations, suitable environmental growth condition, and appropriate level of nutrients. I told you in the beginning itself, the environmental conditions, the microbial microorganisms and the other things are plays a very crucial important in order to achieve the degradation, 100% degradation. That is a main disadvantage. It is difficult to extrapolate from bench to pilot scale studies to full scale field. So what we achieve in the laboratory, so cannot be 100% achieved, can be achieved in the field condition. That is a disadvantage, major drawback. Bioremediation often takes longer than the treatment options. Okay, so these are some of the disadvantages. Okay, not instantaneous, often need to develop system Always need to test optimized conditions empirically, not with computer models. May have inhibitors present. Compounds may not be in a biological form, biodegradable form. So these are some of the disadvantages. Okay. So I'll give you some of the successful stories of this bioremediation process. In the disasters case, studies in jet fuel and Hanson, South Carolina. So here you can see that oil fields of Kuwait, they are successfully cleaned up using this bioremediation process. Okay, in 1989, Exxon vessel an oil spill during cleanup. Researchers learn from about the naturally occurring ways that bacteria convert into harmless compounds. So this you can see that some of the successful stories I give you. Okay, in South Alaska, okay, Central Alaska, they have carried out this process. Okay, and then so this is about the Alaskan bioremediation project. It started in 1989 in King Island. So that is carried out very successfully. Okay, they got very positive result. Approximately after 10 to 14 days, visual reduction in the amount of oil converting into rocks or apparent. Okay. So these are the applications based on the promising result in the field. Future strategies for transition bioremediation, recovering of valuable metals, bioremediation of radioactive waste. So these are the future strategies and cleaning challenges for bioremediation. Okay. So finally, I would like to conclude bioremediation is a simple natural process which is less expensive than the other technologies that are used for cleanup of hazardous waste. Bioremediation can also be carried out on site, often without causing major disruption of the normal activities. It's an environmental friendly approach. Okay, thank you. Okay, so this is about my lecture. So even though my major thing is bioremediation, that is use of microorganisms or plants in order to control the pollution in the environment. So, okay, any questions?